The broadcast is now starting. All attendees are in listen-only mode.
Welcome. Happy New Year, everyone. This is Vicki Binger coming to you and welcoming you to CAC2's January all-member webinar entitled The Blood-Brain Barrier, What If You Could Get Drugs Across? Before we get started and I introduce our presenters today, I'd like to do a small amount of housekeeping. The main thing is I'd like you to look out for the February announcements, which will be coming out next week. It will be full of goodies. We have been working on a proceedings document from the research conference, and I think you'll be very pleased to receive that. We'll have information about our February webinar, which will be, I believe, February 16th, but we're finalizing the details on that. I'm hoping to have the annual meeting save the date information for our June meeting, and an invitation if you have been thinking about being interested in serving on the board. There will be a little blurb about that. So right now, as always, your line is muted to eliminate background noise and telephones and so on, but you have the capability to raise your virtual hand on your GoToWebinar control panel if you have a question, and Trish Krieger will call on you. You can also submit questions through the control panel by typing them out. What I understand is that today our presentation will leave ample time at the end of the presentation for conversation about this topic, but our presenters have asked us that if you do have a point of clarification or a question as they're going, they would be glad to take care of that question on the fly as they go. Rodeman, if you would just put one slide forward to the CAC2 values slide. This is the place that we always start when we come together, and that is our reminder to put the children and their families first in everything that we do, and our other values that help us support each other and be accountable and efficient and inclusive, and really work to give voice to the community while we do the things that we need to be doing. Our presentation today, as you read when you signed up for it, is about a particular technology that this company, Vesalon, has been developing to try to provide a way to move drugs and other therapeutics across the blood-brain barrier. And I'd like to introduce to you Clay Larson, who's the president, CEO, and co-founder of Vesalon. He's a senior executive with a history of medical technology and clinical innovation, including the launch of a new picture archiving and communication business at Fujifilm, and did amazing things with that, growing the unit up to 150 million annual worldwide revenues in just 6 years. And prior to working at Fujifilm, Mr. Larson was a senior executive in the ultrasound and nuclear medicine field. He's going to help with the presentation, but the primary deliverer of our presentation today is Rodeman Lee, who's the founder and executive vice president for strategy and finance at Vesalon, and is a serial medical technology entrepreneur, and was co-founder and senior executive of a venture-backed orthopedics device company, Lee Medical Technologies, which was subsequently acquired. And Mr. Lee has also worked in both healthcare, private equity, and in investment banking. As always, we have Trish Krieger from the Jeff Gordon Children's Foundation driving our technology. We're recording this presentation, and when it is available, we'll post it up to the CAC2 website for you to be able to review later and go back. Without any more delays, I'd like to turn this show over to Rodeman Lee and to Clay Larson and let them do what they're here for. Thank you very much for being here.
Thank you, Vicki and Trish. This is Rodeman Lee, and also thank you to CAC2 and the audience for allowing us to have a conversation about an important topic, which is brain disease. At Vesalon, we're learning that pediatric patients can't be treated in the same way as adults. The pathophysiology of their diseases is different. The long-term consequences of treatment may be an issue for survivors, and the treatment tolerability of toxicity, pain, and recovery is also a factor that's even more important for pediatric patients. The body's natural defense mechanism, the BBB, keeps toxins and drugs from entering the brain. Assuming scientists know what targets to attack with current or experimental therapies, the drugs need to get across the BBB in sufficient quantities and specific locations in order to provide therapeutic effect. So the big question is, how can we treat brain disease if we can't get the therapeutics there in a targeted way? This is an obstacle not only for pediatric brain cancer, but for pediatric patients afflicted with certain rare and orphan diseases, as well as adults with neurodegenerative disease. Our approach is to use non-invasive ultrasound and microbubbles to enhance the delivery of small or large molecules directly into the target in the brain without having to change the molecule in any way. We use two well-understood components, non-invasive ultrasound and microbubbles, in a new way to safely and transiently open the BBB. Microbubbles are smaller than red blood cells, and as a blood pool agent, they travel throughout the vasculature, even into tiny capillaries. They are FDA approved for diagnostics and are very safe to use in the human body. There are only 4 FDA-approved microbubbles products in the US. All of them are for diagnostics. The gas within the microbubbles reflect the ultrasound signal, thereby amplifying the image for diagnostics. They're used as a contrast agent mainly to enhance imaging of the left ventricle in echocardiography. Our microbubbles have a lipid shell and contain an inert perfluorocarbon gas. They come in a vial in the form of dry powder, are mixed with sterile saline, whereupon the microbubbles are reconstituted and are suspended in a solution ready for injection. Microbubbles have a half-life in the body of roughly 8 to 10 minutes, where the gas eventually diffuses out of the shell and is excreted through the lungs, while the lipid shell is processed by the liver. A little technical difficulty there, sorry. Unlike in diagnostic imaging, we're using the microbubbles not for their reflective properties, but for their mechanical properties. With focused ultrasound, when the ultrasound is aimed at the target tissue and the microbubbles cross into the ultrasound beam, the microbubbles undergo rapid oscillation, known as cavitation, meaning the microbubbles expand and contract repeatedly and rapidly. This mechanical cavitation is what essentially opens the blood-brain barrier. Here's an animation showing how the blood-brain barrier is opened. The smaller translucent microbubbles are shown alongside red blood cells, and drug molecules, which are the tiny red dots, are also co-administered. What you'll see, hopefully, is that when the microbubbles pass through the region of the ultrasound beam, the microbubbles cavitate, thereby loosening the tight junctions to allow drug molecules, which are the small red dots, to extravasate directly into the brain target. Extensive preclinical testing with mice, rats, and monkeys have shown that use of ultrasound and microbubbles is safe for both short and long-term exposure to ultrasound and microbubbles, and it's also effective in delivering small and large molecules into the target. The BBB has been shown to remain open for a limited period of time, generally a few hours, and the size of the opening can be controlled. Clinicians recognize this technology and are trying to move this approach into patients. In fact, 20 patients with glioblastoma have been treated with ultrasound and microbubbles to open the BBB to enhance delivery of chemo in 2 safety studies. One is a French study which uses a surgical burr hole to implant an ultrasound transducer, while a second, a Canadian study, uses an MR-guided helmet with over 1,000 transducers that was originally designed and approved for uterine fibroid ablation. Our approach is specifically designed to open the BBB, which can be done in a roughly 1-minute procedure in an outpatient setting. Also, our approach will be used next year in an Alzheimer's disease clinical safety study. Our approach is based on over 12 years of research and more than $11 million of NIH funding. Essentially, we're commercializing technology that's been developed at Columbia University, which is a leading institution in the field of opening the blood-brain barrier with ultrasound and microbubbles. We have a comprehensive system of microbubbles, transducers, and software that's been extensively tested and we believe is ready to begin human use. After planning the treatment, the patient setup should take just a few minutes. The transducer needs to be placed on the patient's head using software or 3D tracking to assure the beam is aimed accurately. The actual treatment to open the BBB should take less than a minute, allowing the BBB to remain open again for just a few hours. Depending on the route of drug administration, our system would be used to time the opening and its duration to coincide with the optimal drug concentration in the patient. The opportunity to treat brain disease is vast and is what drives us to solve the problem. Experimental approaches, such as direct injection of drugs into the tumor via surgical procedures, are being tried. Device companies have catheters and other tools used for CED. Pharma and biotech companies are trying to modify or attach their molecules to other agents, but this will involve extensive safety and efficacy studies, adding years and many millions of dollars to the process. Other component vendors do not have a total solution, nor do they have the IP, the intellectual property. We can target and enhance the delivery of drugs into the brain. We believe that there is an accelerated way to get our novel approach into clinical use via the FDA Humanitarian Device Exemption Program, HDE. Our initial indication is pediatric malignant brain tumors. Rather than an invasive surgical approach, we would use our non-invasive solution to show, first and foremost, that it's safe. Under the HDE program, as long as we show safety and probable benefit, FDA will grant approval to commercialize our solution. In our safety study, we're proposing to FDA that we allow the clinician to use drugs they see fit to treat DIPG. We would also propose to use our system to enhance drug delivery for other brain cancers. But we need to show FDA that our total patient population is less than 8,000 patients and that could be treated using our device in order to qualify for the HDE pathway. So our mission is to change both the outcome and quality of life for patients, especially the young and old, suffering from brain cancer and other neurological diseases. And with that, we want to thank you again for attending and participating, and we open it up to any questions and comments that you may have.
Hi, everyone on the call. If you have a question, please just click your raised hand icon so I'm going to call on you and open up your microphone.
In the meantime, Rotem and Mr. Larson, one of the concerns that we have that you address pretty well, but you may want to reiterate, has to do with maybe what a layman would misunderstand as microbubbles in the bloodstream, and what effect that would have kind of immediately, and then the possibility for accidents having these microbubbles floating around, even if it is for 8 to 10 minutes. And I know you and I have talked about that a little bit, but could you just kind of talk to a layman's audience about why you're really feeling very comfortable about the safety profile of this approach?
Clay, why don't you take that?
Sure. This is Clay Larson. I think if you look at microbubbles, a lot of people have not heard about them before, but they're in a kind of a class of agents that are used typically in imaging departments, radiology and cardiology, that are termed contrast agents. So if you have an X-ray or a CT scan, they use iodine-based contrast agents for the X-rays. If you have an MR exam, they use something called gadolinium-based contrast agents. And for ultrasound, they use microbubbles. If you look at the safety profile of microbubbles, it has by far the highest safety profile of any of the other contrast agents that are used in imaging. And currently they use this very commonly with sick cardiac patients. So they're giving these to sick cardiac patients, as Rotem had showed. It helps light up the left ventricle so you can see difficult to image patients. But when you inject the microbubbles, it's referred to as a blood pool agent, meaning it goes everywhere in the body that red blood cells go. That's why they're actually somewhat smaller than red blood cells. So wherever there's even the smallest capillaries with red blood cells, the microbubbles go through there just as easily. So it's a very innocuous contrast agent that really doesn't do anything in the body unless you actually start focusing some ultrasound on it. So it's extremely safe, and typically, because it's been used in adults for cardiac, it has not been used as much in pediatric applications, although there was just an approval by another microbubble company for a pediatric application for helping look at urinary tract issues. So these microbubbles have had experience in pediatrics and are extremely safe compared to any other contrast agents that are used in imaging.
I would also add that the microbubbles, sometimes when we describe our approach, some people may be left with the impression that the microbubbles actually cross the blood-brain barrier as well, but they do not actually cross it. As Clay mentioned, they stay in the vasculature, but what they're doing by massaging the endothelial walls through the cavitation is allowing small particles, and small is a relative term because we can allow for both small and large molecule drugs to cross the BBB. So the microbubbles themselves stay within the vasculature and again are processed and excreted through the lungs and liver.
Well, that's very helpful. Trish, have we had any of our listeners pose questions or raise their hand?
Yes, we do, actually. Amy Weinstein, I'm going to turn your microphone oh, we just lost Amy's microphone for a brief moment, but she did send me the question as well. So let me read that to you. So Amy asks, are you working to apply this technology in specific medical institutions? And if so, can you share that with us?
Yes, we are. We are in discussions with a major cancer institute. We can't disclose the name of it yet, but it's one of the leading cancer institutions, we believe, in the world, and we are working with them to hopefully have them become or be the lead institute for our clinical safety study for pediatric malignant brain tumors.
Terrific. Amy has a follow-up question. She says, I don't recall seeing this topic being covered at the ISPNO meeting in Liverpool in 2016. Did I miss it? If you were not there, why not?
We were not there ourselves as Vessalon, but there may have been scientists who have had presented at that particular conference or others like it. It's generally the field of focused ultrasound, and I'll take a crack at sort of talking about why we haven't been presenting, and then Clay, you might want to follow up. But we are taking technology that's been done or that's been tested extensively in an academic setting, in a research setting, and as we talked about with the two other companies that are actually pursuing clinical safety studies, that work is really emerging, and so there are a bunch of other factors involved, but we think that we're actually uniquely positioned because we own an FDA-approved microbubble, and we're the only company that is pursuing a new therapeutic indication for the microbubble. We believe that that's been one of the limiting factors for why it hasn't been pushed out to the clinic or commercially until now.
Thank you very much. Our next question comes from Maureen Lilly. She asked, will the opening of the Oncology Center of Excellence at the FDA speed up your approvals?
Let me go ahead and take that. In addition with talking with this really world-class cancer institute about what clinical trials we can conduct, we're also in discussions with FDA about the regulatory pathway that we would need to go ahead and go under. And as Rodeman's slides discussed, we are seeking to have this listed under a humanitarian use device, which you then would have a humanitarian device exemption pathway where, again, you just need to demonstrate safety and probable benefit, and you do not need to go ahead through extensive pivotal trials to prove efficacy in order to be able to make this available for patients. The Oncology Center of Excellence is even very new for the people at FDA, so they kind of have know about it, but the only thing that there's no specific people on the staff in that center that have their contacts directly available. We've reached out through both the Orphan Products Division and we have some other contacts in the rare drugs program to see if we can get some connections there, because if you look at the objectives of this Oncology Center of Excellence in the FDA, it seems like it would be right up our alley. But some of the documentation that they have for the center still says you have to kind of go through all the same processes and standards that the other centers within the FDA require. So I don't know if it would necessarily be able to help us obviate any steps, but I think it would help us perhaps get a little higher level focus and hopefully potentially a little bit faster review process as we're going through the standard steps.
Thank you very much. Our next question comes from Greg Awney. Go ahead, Greg.
Can you guys hear me?
We can.
Okay, good. I just had a question. The preclinical mouse models, have you actually given chemotherapy along with the microbubbles and the focused ultrasound? And if so, what chemotherapy agents were delivered and what kind of toxicity did you see in the animals?
Yeah, there was one study. These are not studies that we have necessarily conducted because there's a body of literature that we can leverage when we go to the FDA, because they allow other types of studies to be submitted as evidence. There's one group that actually treated mouse xenograft tumors with bevacizumab and Avastin, and was able to demonstrate at 2 different levels of acoustic energy, 1 level of acoustic energy. These are all with microbubbles, of course. One kind of very modest low level of acoustic energy was able to get 5.7 times as much Avastin into the tumor, and at slightly higher levels of acoustic energy, which is all still very much within the safe levels for human use. They were able to increase that to 58 times. That's 58-fold, not 58%. So it is quite a dramatic increase in drugs that's actually been being delivered into the tumor itself.
And those were, you said xenograft, those were animals that had flank tumors. Those were animals that actually had intracranial tumors where you're delivering?
Well, they would have an implanted set of tumor cells that are allowed to grow inside the mouse.
In the brain, though, so an orthotopic model then, right?
No, I'd have to relook at this, but that particular one that I was referring to was in Taiwan. So I'd have to look at specifically exactly, where they had implanted it.
Is the idea that it could also be used to deliver drugs to, you're starting with brain tumors, but could it be used to deliver drugs preferentially to solid tumors with the same technology by disrupting blood vessel integrity?
Yeah, the same process would be able to work really in any tissue type, including solid tumors, and in fact, the same effect, interestingly enough, can actually work transdermally. So there's some very interesting capabilities. It's an extremely powerful platform for helping deliver these. So certainly solid tumors, tumors in different types of the body are all candidates for this type of, in a way, it's kind of a pre-treatment. In other words, you're pre-treating the tissue so that then whatever systemically administered agent can get to that location. It's also very interesting that it's been shown, again, this is still in an animal model, that for the, when Rodeman showed the slide of other competitive ways, you saw the one where inserting a catheter and actually using a convection-enhanced delivery, delivering a drug under pressure. They've actually had problems with the drug diffusing far enough away from the needle tip to be effective. What Columbia has actually shown in a mouse model, that if you pre-treat that same tissue and then do the same infusion, that it substantially increases how far the drug diffuses away from the needle tip. So, so that's an interesting alternative. So this is not necessarily going to be limited only to systemically administered drugs. And then what's even equally exciting is that it has also been shown to have an effect that pre-treating tumors with ultrasound and microbubbles can act as what's called a radiosensitizer, in that it would actually make those tumor vessels, tumor tissues that are exposed to ultrasound and microbubbles, it actually makes them more prone to cell death after radiation therapy. So that has a lot less data to support it, but it's very, very strong data that shows that that's another, opportunity. So we really are looking at a very much of a platform technology that may have lots of applications.
Yeah, thank you. It's a fascinating technology.
Thank you, Greg. Our next question will come from Amanda. Amanda Griffin, I'm going to open your mic now. Go ahead, Amanda.
Thank you, Trish. I would like to find out whether you can elaborate any further on panobinostat for the treatment of DIPG. Sorry, I kind of missed the slideshow a bit when you were speaking about that. But I think what I heard and seen was that you were looking at panobinostat as a single agent treatment for DIPG using your technology. Is there any more information that you can provide about that?
Yeah, that is one both tumor type and drug combination that we are extremely interested in. Yeah. With panobinostat, you may be familiar that some of the drugs have different sizes, and as we mentioned in some of the slides, we actually have the ability to deliver both small to very large drugs. So we can deliver smaller chemo agents like panobinostat, even up to larger monoclonal antibodies like Avastin. So size is not a limitation for our technology. One thing that it has been demonstrated in lots of the preclinical work is that there is a little bit different percentage uptake between gray matter tissue and white matter in the brain. And this is due to the vascular pattern to it. And so gray matter would maybe have about 3 times as much uptake after being treated with ultrasound microbubbles than white matter. And so we believe the brainstem, the pons where DIPG is, would act much more like gray matter from that standpoint. So we're looking at structuring clinical studies where a DIPG protocol may have a slightly different settings as opposed to, let's say, an astrocytoma in the cortex, just to make sure that we're getting the right amount of penetration of the panobinostat into the actual tumor.
Okay, thanks for that. And can I ask, and you've probably explained this already, but are you able to measure, the amount of drug that's actually, getting into the brainstem, and how much of it? Is that something that you're able to measure with the, with your drug delivery system?
Yeah, it's a fantastic question, and one of which we've talked with our clinical partners quite a bit about. When you look at, how do you go ahead and tell whether or not the blood-brain barrier has been opened? Oftentimes you use a couple of different techniques, some of which are proxies for what's being penetrated. Some are direct measurements. Obviously, a direct measurement, if you're doing a mouse model, would actually be, actually taking part of that tissue and measuring if the drug has actually gotten into the tissue. Oftentimes, imaging using an MR contrast agent. And the MR contrast agent, which is based on gadolinium, actually is very similar in size to panobinostat. So if you go ahead and typically a DIPG tumor does not take up contrast, especially initially upon diagnosis and before it's been treated with radiation or anything. So early in the process, we would be able to show, through an MR proxy that we're able to open up in the tumor so that MR contrast agent can get in, which would then mean panobinostat would also be able to get in. We've talked to this institution about doing some, pretty advanced things using positron emission tomography tracers, basically radio labeling radio labeling panobinostat with, let's say, carbon-11, so that you have a definitive imaging-based measure to see that you're actually getting the actual drug into the tumor. So we have talked about structuring a future trial along those bases. So clearly that would be the gold standard, that yes, you're getting the actual drug in, but certainly there's other proxies like MR contrast, which would be very good for panobinostat because it's the same size. If you were trying to deliver Avastin, which is a much bigger molecule, there could then be the argument, well, yeah, you're letting in the gadolinium, but Avastin is like, five times bigger, so there would be some questions about, efficacy there.
Yeah. Oh, thanks very much for that, and I'm just really excited about the, everything that's been explained. Obviously, DIPG, there's been a lot of, talk about whether, the drugs in all the trials that have been conducted so far, whether the drugs are actually getting into passing the blood-brain barrier. So, yeah, it's very, very exciting. Thanks very much.
Yes.
And excuse my accent, I'm Scottish.
This is Vicki, and I have a follow-on question to Amanda's first question when she was referring to the slide that matched the various indications with particular drugs. As it relates to the third bullet on your discussion question, and which is how do we get drug company buy-in, tell me a little bit about your experience, and are you facing reluctance from the agent providers to partner with you to do these trials, and what would be the source of their reluctance? Why would they not want to participate and provide the drug?
Yes, Vicki, this is Rodeman. Thank you. That's a great question. We are not finding reluctance, but what we are finding is that it is somewhat difficult to find the decision makers within the drug companies that have an interest in specifically pediatric brain tumor treatment or drugs. And so we have and are continuing to approach people in the business development side of the drug companies and trying to get to them or get in front of them with our approach, and then navigate within the drug company bureaucracy, if you will, who the right people are that would be willing to consider a collaboration. So any assistance that you and your group may have that would identify the right people to talk to within the pharma or biotech companies, we would love to work with you on that.
Yeah, this is Clay. I think I think that once we kind of finalize, let's say, a clinical trial protocol, especially with the level of medical centers that I think are going to be interested in working with us, I think the access to getting, let's say, the drugs, which are obviously very expensive drugs, getting them available for the trial itself is not as much of an issue. I think, we think that we'll be able to get, cross that bridge when we get to it. I think it's, getting interest from these drug companies to maybe assist us from early stage investment standpoint so that we can accelerate both the development and the pathway through the regulatory process because they're familiar with obviously drugs, but they're not as familiar nor as interested in devices. And we kind of fall into the category of a device-drug combination because we have our ultrasound device, but we also have our own, drug, which is the contrast agent is considered as a drug. So I think it's more getting their interest in our whole approach rather than necessarily just providing the drugs for our clinical trial.
I see. Well, thank you for that. I was trying to imagine why they would not want to participate, and it does seem more like an information highway kind of thing than really a danger or risk kind of situation.
Yeah, it's certainly not a danger or a risk, especially I think the huge benefit of our approach is the fact that there is no need to actually change either the formulation of the drug nor even the administration of it. So in other words, if a drug is already set to be an oral tablet or capsule, that's fine. If it's set to be an injection, that's fine. If it's set to be an IV drip, that's fine with us. We're kind of an independent agent that again kind of pre-treats the tissue to be more absorbent, if you will, to these systemically administered drugs. So again, we're not looking for collaboration to modify the drugs. So we actually feel that with all the different drugs that have been tried in a number of these different disease processes, we would hope to get some more interest from the drug companies to help potentially revive some previously unsuccessful drugs whose bioavailability was thwarted by the blood-brain barrier. So we also think that that's a potential huge opportunity in the future.
Great, thank you both. We do have another question here from Janet. Janet, I'm about to open your microphone. Go ahead, Janet.
Yes, can you hear me?
Yes, we can.
Okay, great. I had a question for you about, of course, my focus is generally DIPG, and I know that some DIPGs are come back testing as astrocytoma, some come back as glioblastoma multiforme, and of course it's all location, location, location. But and so far as the panobinostat, I heard you mention something about combination therapy, and I was wondering, are you working with the greater COG community in their combination therapy project for DIPG? I know specifically Dr. Charles Keller at the James Cancer Therapy Development Institute.
We are not working with Dr. Keller specifically, but we certainly would be open to it. We are concentrating our efforts at this one cancer center, this major cancer center, just because we are, as you might appreciate, as an emerging company, we're very focused on our plan, and we have limited bandwidth as well as resources, but limited bandwidth more particularly, and so we're trying to we're trying to work with institutions and do so in a way that will accelerate the process. And so the simple answer is we have not reached out to a lot of people, but
Sure.
we are trying to selectively reach out, if that makes any sense.
Of course, and it seems like with this technology, just honing it and getting it to a point where it can be usable, and then communicate potentially with these other scientists about combination therapy, because they're finding that the reason why I brought it up is because my organization is promoting and to a certain extent another piece of biotechnology that that should help in the determining of what particular drug combination would be good per patient. So this is much further down the road then, I guess, but I just wanted to let you know I'm really, really excited about what you're doing, and I came on late as well, and I'm wondering, will there be some sort of presentation that we can have after this to refer to with all the information you've given?
Yes, we're very happy to provide copies of our slides to anyone who would like them.
Janet, as soon as Trish and I can get this entire webinar processed and posted up, I'll link it to the CAC2 website, and it will be available for you to listen to the entire recording, barring any technical difficulties that we may have had. Sometimes we do have audio difficulties, but we've tested it, and we believe we'll have the full recording of this. And I will, in addition to putting it on the website, we'll make sure to put that link in the February announcements when I send them out next week, so that you can reference it as you need to, and you can see the whole thing. For people like Amanda in Australia, who had to get up in the wee hours of the morning, and for other folks that work during the day and can't be here on the call, we want to make sure that we share the information.
Off the top of my head, with referring to the difficulty in getting the information to certain company heads that aren't necessarily focused on the pediatric neuro-oncology needs in the community. Genentech came to mind, ABC Squared and NBTS came to mind as excellent resources for perhaps sourcing those people. That was just something I wanted to say off the top of my head.
Yeah, that's great, Jan. This is Clay. Yeah, any of those connections would certainly be helpful. I just want to make another comment when you're talking about combination therapy, on a couple of points. First of all, whatever combinations of drugs would be helpful, our kind of platform would be able to help. Let's say it's a combination of 2 or 3 different drugs. It would be able to help all 3 of them access the tumor better. One problem, as you may be aware, is that some of these tumors can have heterogeneous parts to them between both inner and outer parts. Again, it's been shown that the ultrasound and microbubbles should be able to allow better absorption, across these different in other words, it would increase the permeability across all regions, relatively increase from what the baseline is. So that's also very very, very promising. In talking with our clinical partners too, this is the other tricky thing we have in terms of setting up a clinical trial from a clinic standpoint, but also having the proof for FDA. So we're literally going through right now, do we pick one tumor with one drug, or what would be more of our preference is, do we go ahead and work with maybe more of a general class of tumors like pediatric malignant brain cancer, and then have our tool be consistently used in the treatment, but let the physicians choose whatever they feel is the best care from a chemotherapy standpoint. So in other words, if it's going to be a mixture of Temodar and CCNU, or any combinations of those, we would prefer to kind of have us be almost like an agnostic tool for the physicians to use whatever combinations they would like, and for us to then present that total information to the FDA, as opposed to just do a rifle shot with one tumor type with one drug.
Right. Well, thank you so much.
Gentlemen, most of our questions are really around exactly that, what Janet was asking, and it is really about what organizations could do to get more information, get more involved, assist you in moving the project forward. So, so we really appreciate that. If there's anything additional to add, now would be a great time. The balance of our questions, as I said, are related to that exactly. Hello?
So Rodeman and Clay, I guess that's what I was going to ask you. If you could put your contact information up, and you know, with regard to those issues, I think that that our members are spread out across the United States and even in some international settings, and they do have contacts, probably more with clinical settings than they do with companies, but they are oftentimes pretty informed about what the current practices are and what might be a good place. So if you have anything to add about the way our members can support the effort to speed what you're trying to do, or they can address questions to you directly if you would like to speak to that. Otherwise, I'll leave you to kind of conclude your presentation, and at the very end, I'll just come back on and thank you and thank everybody who participated in the webinar today, and focus our attention to what actions we can take and move on from there.
Thank you, Vicki. This is Rodeman. Just to kind of recap, we really look at this as an opportunity to collaborate with many stakeholders. We would welcome the interaction and discussion with your group and your members, just because you can add such valuable insight to not only the disease but to treatment issues that ultimately will make our solution effective for these patients, and that's what really drives us. So we're very open to questions, comments, criticisms. We want this to be tested by everyone to make sure that when we do use our system in patients, that we have the very best solution possible. So we welcome all comments and questions and suggestions.
So I'd really like to thank you very much for proactively seeking us out and for the work that you're doing on behalf of our particular patients and for patients with other diseases that are going to benefit from your system. And I will endeavor to get this posted up. And folks, thank you for your time on this Thursday and for making the switch from Wednesday to Thursday. I know that seemed to have tripped up some people that didn't make it on, but I appreciate your time, and we will look for an email from me next week with all the February announcements. And other than that, Trish, if you have nothing else, and Clay and Rodeman, thank you very, very much for presenting to us today, and to CAC2 members. This will be available for preview and for review as soon as we can get it up.
All right, thank you.